Imagine walking into a massive library that contains the entire instruction manual for a human being. Volumes upon volumes of genetic code. Now, imagine you walk over to the section labeled Cancer, pull a random book off the shelf, and just open it up. What do you actually see?
You see chaos.
Yeah.
Honestly, you see 10 million typos.
Yeah.
I mean, scribbles in the margins, torn pages, entire sentences that have just been rewritten backwards.
Right. Exactly. And that is the staggering reality we are dealing with today. The Cancer Genome Atlas, or TCGA, contains over 10 million distinct mutations. But here is the statistic that completely floored me when we were prepping for this deep dive. Out of those 10 million typos, for the vast majority of them, we have absolutely no idea what they actually do.
Yeah, it's the dark matter of oncology. In the field, we call them VUFS, which stands for Variants of Unknown Functional Significance, which is really just a very polite scientific way of saying, well, we know the code is broken, but we have no clue if it actually matters.
And that's the nightmare scenario for a patient, right? You get your genome sequenced, the doctor points to a mutation and says, look, we see it, but we don't know if it's just a harmless passenger tagging along for the ride or if it's a silent killer that's actively driving your tumor.
Precisely. Because if we treat a neutral passenger mutation with highly toxic drugs, we hurt the patient for no reason.
Mm-hmm.
But on the flip side, if we ignore a mutation because we don't recognize it and it turns out to be a driver,
we miss the chance to save them.
Exactly. We miss the window.
So today we are diving deep into a recent paper from Nature Genetics that proposes a really brilliant solution to this exact problem. The paper is titled Pan-Cancer Inference and Validation of Hypermorphic, Hypomorphic, and Neomorphic Mutations. We're going to break down every concept, every algorithm, until the whole picture makes sense.
It's a dense title, I know, but the solution they present, a computational framework called PHNToM, is incredibly elegant. It essentially promises to turn those millions of unknowns into knowns without needing, decades of expensive lab work for every single variant.
It's basically an intelligence gathering mission for our DNA. But before we get into the how, and the tech here is fascinating, we really have to understand the what, because this paper fundamentally redefines the villains of the story.
Right. For a long time, people thought of cancer mutations in a very binary way. Is the gene on or off? Is it a driver or a passenger?
Mm-hmm.
But biology is much, much messier than a simple light switch. This paper argues we need to look at 4 distinct personality types for these mutations.
I love this part. It feels like we're profiling suspects in a crime drama. So for everyone listening, let's unpack the first 2, which are probably the most familiar. We have the hypermorph and the hypomorph.
These are the classic bad guys. Let's start with the hypermorph. This is a gain-of-function, or GOF, mutation. Imagine a normal protein that's supposed to send a polite signal to the cell saying, hey, time to grow. A hypermorph is like that same protein getting stuck with a megaphone screaming, grow, oh God, 24/7. It works too hard. It's completely overactive.
The toxic overachiever, got it. And the hypomorph.
That's the loss of function, or LOF. The mutation basically breaks the protein. It's supposed to do a critical job, maybe repair damaged DNA or tell a cell to stop dividing, but it just clocks out. It's effectively dead weight.
Okay, so one does too much, one does too little. Then we have the third character, the isomorph.
The neutral mutation, the innocent bystander or passenger. The genetic code is technically changed, so it looks different under a microscope, but the protein still folds correctly and does its normal job. It's totally harmless.
And identifying those isomorphs is huge because, as you mentioned, you don't want to nuke a patient with chemo for a passenger who isn't even driving the car.
Precisely. But then we have the fourth character, and this is the one this paper is really obsessed with. The neomorph.
The neomorph. I mean, this sounds like a sci-fi villain.
It really is the most dangerous and tricky category to nail down. A neomorphic mutation is a gain of new function. The protein doesn't just work faster or slower, it goes completely rogue. It starts doing things it was never, ever designed to do.
The paper gave a really specific example of this that I want to drill into for our listeners, the SMAD4 mutation. Let's use this as our case study.
Yes, SMAD4 G386D. This is a perfect illustration. So normally SMAD4 is a standard signaling protein. It has a very specific, well-regulated job description in the cell. But this specific mutation, G386D, causes it to form a weird, aberrant relationship with another protein called GSK3B.
Like a bad workplace romance?
That is exactly what it is. In a healthy cell, SMAD4 and GSK3B should not be interacting like that. They belong in different departments. But because of this illicit affair, the mutated SMAD actually gets incorrectly phosphorylated, and it messes up the entire 1 beta-catenin pathway.
And for those who might not know, the 1-beta-catenin pathway is a massive driver of cell proliferation in cancer when it gets out of control.
Huge driver. So think about the clinical implication here. If you treated that patient with a drug designed for a broken SMA4, a loss of function, it wouldn't work at all.
Because the protein isn't broken. It's doing something entirely new. It has a completely new agenda.
Exactly. It would fail completely. Yeah. And this actually leads us to another core concept the paper highlights that I found deeply unsettling: the field effect.
Right, because we usually picture cancer starting with one big bad mutation, like a bomb going off in the cell.
That's the Big Bang theory of cancer, which is popular but often incomplete. The authors argue that tumorigenesis, the actual birth of a tumor, is frequently a field effect. It's not one big explosion. It's the slow accumulation of multiple weak events.
So maybe 3 or 4 of these unknown mutations, these VUFS, are actually cooperating in the background.
Yes. Individually, they are weak. You look at them on a genomic screen and think, harmless passenger. But together, they tip the scale. They create a field of cellular instability that allows the cancer to finally take root.
Which makes characterizing these unknowns even more critical. You can't just hunt the big game anymore. You have to understand the swarm. Okay, let's do a quick recap because we're about 5 minutes in and I want to make sure we're all on the same page. We are dealing with millions of variants of unknown functional significance, VUFS. They can be lazy, which is loss of function, hyperactive, gain of function, neutral passengers, or rogue agents, the neomorphs, and we absolutely need to sort them out because tumors can be built from a quiet collection of these weaker, unknown signals.
And that brings us to the weapon of choice for this entire operation: PHNToM.
PHNToM, which stands for Protein Activity-Based Identification of Hypermorphic, Neomorphic Effectors. I am so glad they just went with the acronym. So how does this computational framework actually work? It uses something called VIPER, right?
Yes, the VIPER algorithm. This is the core technology that makes everything else possible. And to really grasp a VIPER, you have to flip your traditional thinking. Older methods look at the mutation itself. They look at the physical shape of the mutated protein. VIPER doesn't care about the shape. It only cares about the influence.
This goes back to the intelligence gathering analogy you mentioned earlier.
Right. Imagine a protein, let's call it protein X, is a general in an army. You want to know if that general is actively shouting orders to attack, but you can't actually see the general. He's locked in a bunker. However, you can see his soldiers, the downstream genes that he specifically controls.
So if you look out and see the soldiers are marching, you know for a fact the general is active inside the bunker.
Exactly. VIPER looks at the regulon. That's the specific network of target genes regulated by a protein. If those target genes are being heavily expressed, VIPER infers that the protein is active. It doesn't look at the mutation. It looks at the functional consequence of the mutation on the rest of the cell.
That is so incredibly clever. It literally judges the tree by its fruit. Yeah. But how does it know which soldiers belong to which general?
That's where another algorithm called ARATION comes in. ARATION maps those connections. But it requires over 100 distinct gene expression profiles to build a reliable, statistically confident map of that network. Once it has that map, PHNToM can start its main workflow. It compares the unknown mystery mutation against a known established mutation.
Okay, so it compares the mystery suspect against a known criminal profile.
Yes, but to do that accurately, you need a pristine control group. And this is where the paper highlights a massive headache in cancer research, defining what is actually normal.
I would assume normal is just the wild type, the non-mutated version of the gene.
You would think so.
Yeah.
But the authors point out that even wild-type samples sitting in a tumor database can act incredibly weird. Maybe there's an upstream mutation in a different gene affecting them. Maybe there are paracrine signals, like inflammatory chemicals from nearby cells altering their behavior. If your normal control is actually acting like a stressed or cancerous cell, your entire comparison is garbage.
So how did the researchers fix that?
They went outside the tumor databases. They went to the GTEC-X database, the Genotype Tissue Expression Project, which contains pristine data from genuinely healthy human tissue. They filtered all the tumor samples labeled wild type and literally threw out any sample that didn't statistically match the truly healthy tissue from GTEx. They called this highly refined, purified group PWT0.
PWT0, the pure wild type.
Basically. But they didn't stop there. Because cancer isn't just one monolithic thing. Breast cancer, for example, has very distinct subtypes: luminal, basal, HER2 positive. A mutation might look super active simply because it happens to be sitting in a luminal subtype where that particular pathway is naturally busy anyway.
So you could easily mistake a simple background subtype difference for a dangerous mutation difference.
Exactly. Background bias is the enemy of this kind of research. So they used a key nearest neighbors approach. For every single mutated sample they wanted to analyze, they didn't just compare it to a giant generic pool of normal samples. They set K to 5, meaning they picked the 5 specific normal samples that were most similar to that patient's tumor type in every other way, except for the mutation.
That is incredibly meticulous. It's like finding 5 twins who live the exact same lifestyle, eat the same food, but just don't have the mutation, and comparing your suspect only to them.
It is meticulous, and that level of mathematical rigor is exactly why they believe their predictions hold up so well in the lab. They aren't being fooled by the noisy background chatter of the tumor.
So we have Viper looking at the soldiers, Ericsson building the map, and we have this rigorous K-nearest neighbors control selection. Let's create another checkpoint here for you listening. We're about 10 minutes in.
The summary of the mechanism so far. PHNToM uses the footprint of gene expression, what the protein actually makes the cell do to judge a mutation, and it meticulously curates its control groups using healthy GTEx data to ensure it's not being fooled by tumor subtypes or noisy fake normal samples.
All right, now we get to the brain of the operation, the logic engine. This is where I saw the acronyms DGN and IGN in the paper and admittedly got a little intimidated. But walk us through this because this is the specific math that actually catches the neomorphs.
It's the secret sauce of the whole paper. To find a neomorph, that rogue agent, you need 2 distinct measurements to compare against each other.
Okay, let's break them down. Metric number 1 is DGN, direct gain or loss of function NES.
Think of DGN as asking a very straightforward question. Is this unknown mutation doing what the specific known mutation does?
Okay, so if the known bad guy always orders pizza on a Friday, does our unknown suspect also order pizza on a Friday?
Correct. Specifically, they look at the top 25 genes turned on or off by the known mutation. Does our mystery suspect turn on those exact same 25 genes? If yes, the DGN score is high. It's a direct match.
Simple enough, direct mimicry. Now, metric number 2, IGN, inverse gain or loss of function NES.
This one asks the inverse question. Is the unknown mutation doing something that the known mutation usually does? But it flips the perspective. It looks at the top 25 genes activated by the unknown mutation and checks to see if they appear anywhere in the known mutation signature.
Wait, I have to be honest, isn't that just the exact same thing said backwards?
Not quite. It's a subtle but massive difference. DGN asks, are you mimicking the known profile? IGN asks, is your total profile fully contained within the known profile?
Okay, I need a visual for that. Can you give me an analogy?
Think of a Venn diagram. If you picture a Venn diagram in your head right now, it makes total sense. Circle A is everything the known mutation does. Circle B is everything the unknown mutation does.
Yeah.
If Circle A and Circle B overlap perfectly, both DGN and IGN match, they are identical. It's a standard expected mutation.
Okay, pure overlap, standard profile.
But if the unknown mutation is a neomorph, it starts doing crazy new things. Circle B starts to drift away from Circle A. It starts covering empty white space on the page that Circle A never even touched.
So the scores start to diverge because the unknown is coloring outside the lines?
Exactly. The logic tree the algorithm uses is brilliant in its simplicity. If DGN and IGN match and are both positive, it's a hypermorph, a standard gain of function.
Got it. Okay.
If they match and are both negative, it's a hypomorph, loss of function. But if DGN and IGN are significantly different, meaning the statistical p-value, the absolute difference between them, crosses a certain threshold that triggers the neomorph alarm.
Because it mathematically proves the unknown mutation is turning on genes that the standard mutation never, ever touches.
Yes, it has gone rogue. It is a gain of new function.
That is the big aha moment of the paper for me. It's not just about the overall activity level, it's about the specific pattern of activity diverging from the norm.
And this mathematical distinction is what allowed them to confidently scan the entire cancer genome atlas.
Which brings us to the actual results. And they didn't just test this on 1 or 2 genes. They characterized and get this number, 583,089 individual mutational events.
It's a massive undertaking, a true pan-cancer analysis. And the breakdown of what they found is fascinating. Out of those hundreds of thousands of events, they found about 317,000 gain-of-function events and roughly 166,000 loss-of-function events.
So the vast majority of mutations really are just stuck gas pedals or broken brakes?
Yes, but they also found about 97,000 neutral events, Those are the passengers that we can safely ignore in the clinic. And most critically for this discussion, they identified about 2,300 neomorphs.
2,300 potential rogue agents. I mean, on one hand, that sounds like a very small percentage of the total. But on the other hand, that's 2,300 specific scenarios where standard targeted treatment might completely fail a patient. Let's talk about a specific case study they used to highlight this. They looked really closely at PIK3CA in breast cancer, right?
They did. PIK3CA is a classic, very well-known oncogene. In the breast cancer cohort, the BRCA cohort, they analyzed over 1,000 samples. They found 333 distinct PIK3CA mutations. Now, traditionally in oncology, we just label PIK3CA as a driver and move on. But PHNToM saw incredible nuance.
It found that biological context matters immensely.
Huge context dependency. This is honestly my favorite part of the study. For example, they tracked down a specific mutation, R108H. When that mutation appeared in uterine cancer, PHNToM predicted it to be a neomorph, acting completely rogue. But when that exact same mutation appeared in breast cancer, it just looked like a standard hypermorph.
That is wild. The exact same typo in the instruction manual implies a completely different command depending on which organ the book is currently sitting in.
Precisely. In breast tissue, the local cellular machinery interprets the typo one way. In uterine tissue, surrounded by different proteins and signals, it interprets it differently. That has massive implications for how we design drug trials.
But before we get too excited about all these computer predictions, we have to ask the hard question. Did they actually prove it? Or is this just highly sophisticated computer wizardry?
Right, validation is everything. And they didn't just guess and publish. They took these computationally predicted mutations, like the FGFR2, N549K, and N549D variants, and they put them into actual living cells in a lab.
What kind of cells?
Specifically MCF10A cells, which are a very standard, reliable, immortalized line for modeling breast tissue. Once they introduced the mutations, they ran RNA sequencing on those cells to see what genuinely happened to the gene expression.
The moment of truth. And what were the results?
The accuracy was staggering. For their predictions of gain of function, loss of function, and neutral events, they hit 100% confirmation in their tested subset.
100%. I mean, that almost never happens in biological validation.
It's incredibly rare. It speaks to how rigorous that PWT zero control filtering was. Now, for the neomorphs, it was a bit harder to confirm, which is expected because you're looking for entirely novel pathways. But they still confirmed about 78% of their predicted neomorphs.
Still, a 78% success rate for identifying a completely new, hidden biological function out of the dark matter of the genome is incredibly impressive.
It is. And they went a step further. They specifically showed that those FGFR2 mutations I mentioned actually cause the cells to grow and proliferate completely independently of growth factors, or mitogens. The computer predicted they were dangerous rogue agents, and the Petri dish definitively proved they were driving unregulated growth.
Now, there was one other really weird phenomenon they uncovered with this tool that we have to touch on. They called it mutational mimicry. This sounds to me like cellular identity theft.
It essentially is, because PHNToM looks at the downstream regular on the soldiers instead of the general. It can look at a mutation in gene A and realize that it is producing the exact same VI signature as a totally different mutation in gene B.
So a PIK3CA mutation might look like it's wearing a TP53 mask.
Exactly. Or it implies they are working sequentially in the exact same pathway, or substituting for one another to achieve the same goal for the tumor. The paper explicitly mentions that PIK3CA mutations often mimic the pathway activation of other known drivers. This helps us understand tumor redundancy.
Why does redundancy matter in the clinic?
Because if you give a patient a drug to block gene A, but gene B is already mutated and secretly mimicking gene A's function in the background, your drug might completely fail. The tumor just routes around the roadblock.
It's like playing a deadly game of whack-a-mole, but the mole has a stunt double that looks and acts just like it.
That is a very grim but very accurate way to put it.
Okay, we're coming up on the end of our time here, so let's wrap this up with the big so what for everyone listening. We've moved from basic lists of mutations to deep functional understandings. We can now distinguish broken parts from supercharged parts from completely rogue parts. Why does this deeply matter for the listener, or more importantly, for the patient sitting in the oncologist's office tomorrow?
It all comes down to true precision medicine. Not just guessing, but precision. Look, if a patient had the mutation that PHNToM flags as a definitive neutral passenger, we absolutely shouldn't be giving them toxic targeted therapies. It's not driving the cancer, we're
Yeah.
just causing side effects.
Right, spare them the toxicity.
But if they have a neomorph, that's the critical piece. Standard targeted drugs are often rigidly designed to fit perfectly into the active site of a specific protein shape. If the protein has gone rogue, if it has changed its shape or its interacting partners, like we saw with SM-4, that standard drug might not fit anymore. Or worse, it might bind perfectly but fail to stop the new rogue function.
So we might actually need entirely new classes of drugs specifically engineered for these neomorphs.
Yes, the takeaway is that we need to start testing patients' tumors, not just for the mere presence of a mutation, but for the actual function of that mutation in real time.
I want to leave everyone listening with a provocative thought building on that context dependency you mentioned earlier. You said the exact same mutation could be a standard hypermorph in breast cancer, but a dangerous neomorph in uterine cancer.
I did, and it raises a massive, almost philosophical question for oncology. If context fundamentally changes function, are we supposed to be treating the mutation, or are we supposed to be treating the environment?
Right. I mean, how many harmless passenger mutations are sitting quietly in our bodies right now, just waiting for the wrong tissue context? Maybe a spike in inflammation, maybe just normal aging to suddenly flip and turn into a neomorph.
It completely blurs the line between genetic destiny and environmental trigger.
Yeah.
In a way I don't think we have fully grasped yet as a scientific community.
That is definitely something to mull over. Before we go, I want to invite you, the listener, to check out the show notes for this deep dive. We've included a link to the original paper, but specifically I want you to look at the box plot images. I think it's figure 2 in the source. It visually shows exactly how these 4 mutation types separate out from each other. It's one thing to hear us talk about Venn diagrams. It's another to actually see the data points drifting away from the normal clusters.
I agree. It really makes the math click in your brain when you see it visually mapped out like that.
And I'll leave you all with this final question to chew on. PHNToM takes an unknown variable and figures out its true nature by looking at its impact on its surroundings. Which unknown variable in your own life, or maybe in your own work, would you apply the PHNToM logic to? Are you judging things by what they simply are on the surface, or are you judging them by what they actually make happen in the real world?
Deep thoughts for a deep dive. I like it.
Thanks so much for listening and exploring the dark matter of the genome with us. We'll see you on the next one.
Goodbye, everyone.